Hello and welcome to lecture number 9. I want to introduce you all to my favorite plants. This is a chlorophyllum. It's also called the spider plant because the leaves kind of look like little spider legs, maybe. And this plant's really cool because it's the only plant designated as a super plant by NASA. And the reason it's designated by a super plant is because it's really good at cleaning out nasty particulate in the air. And so it's an air purifier. And it's really easy to propagate. So you see it has this thing sticking off. That's a little projection, a little string, and on it there are little babies. So each one of those little things is a new plant growing off of my original plant. So if you buy one of these plants, you end up getting a whole bunch more for free. So I could snip these off right here, and I could put them in new pots. And then I could have a whole bunch of these, which is totally what I did. And my whole house is full of these, and I love them. Chlorophyllum. Okay, now let's talk about photosynthesis. Photosynthesis is our topic for lecture number 9. And our question today is, how does photosynthesis work? But really, it's going to be a very quick overview of photosynthesis. And then I'm going to have some practice questions and a study tip for you at the end. When you're studying photosynthesis, it's important to know the structure of the chloroplast. That's what we're looking at here. The chloroplast reminds me of the mitochondria because it has 2 membranes and some of the same vocab words. But once we get inside of the inner membrane, this is where all the cool stuff is and where all the new vocab is going to come into play. So if we were to look at the layer right here, that phospholipid bilayer on a thylakoid, we would find some pigments embedded in it. Pigment means color. These are literally colors that pick up energy from the sun, which I just think is so incredible. The most common ones are chlorophyll A and chlorophyll B. You can see their beautiful complex structure there, but there's definitely more. We don't go into a lot of detail in our book, but there's carotenoids. These are orange pigments like carrots. And then we have other ones like phycocyanin, which is a bluish color. It's common in things like blueberries, but that's actually not what gives blueberries their blue color. The blue color in blueberries comes from a different molecule. And each one of these photosynthetic pigments picks up a different wavelength of light. So if you only use chlorophyll A, you would only get your light where the 2 big peaks are. And so instead, if you use a whole bunch of different pigments, you could use more of your light spectrum. If we were to look even closer into that thylakoid membrane, we would see the electron transport chain, similar to in the mitochondria, but this one's called the chloroplast electron transport chain, so I'll always tell you where it is. Then we have photosystem 2 and photosystem 1. This one was discovered first, which is why it got named 1, even though photosystem 2 is used first in the process. Photosystem 2 picks up the energy from light using those photosynthetic pigments. An electron is then released into the electron transport chain. Water splits and donates another electron to replace the one that's been moved into the electron transport chain. As the electron moves through the transport chain, hydrogen ions are pumped into the thylakoid lumen, creating a concentration gradient, just like we did in the mitochondria. In photosystem 1, you're going to use another ounce of energy from light. You're going to excite the electron again, and then waiting to catch it is NADP. Notice that the P is there. In the mitochondria, we had NAD. So in the chloroplast, it's NADP. So P for photosynthesis, if that helps you remember. The NADP is reduced. It grabs that electron from the electron transport chain, and it becomes NADPH, which now is a full taxi ready to take that high energy electron over to the Calvin cycle. All of those hydrogen ions that you have created inside the lumen would like to go with their concentration gradient. They want to go from high concentration to low concentration. And they're going to go through the enzyme ATP synthase, which will take a phosphate, pop it onto ADP, and create ATP. So ATP, super important. We're going to use that as well as the NADPH in the Calvin cycle. Here's an overview of the Calvin cycle. After we have created ATP and NADPH in the light reactions in the thylakoid membrane, those two molecules, these energy molecules, are going to go over to the Calvin cycle, sometimes called the dark reactions because it doesn't have to happen when there's light out. I like calling it the Calvin cycle because dark reaction makes it sound like it can only happen in the dark, which isn't true. They are going to be fixed with carbon dioxide from the air and RUBP to produce 3PG, which is not your end product. Your end product is rotate those numbers around and you get G3P. G3P is what can be turned into a glucose molecule later, or a starch molecule, which is just several glucose molecules together, or you can use it to help make other types of molecules to build your plants. Remember, this is a cycle, so we're going to keep replenishing Rubisco so we can do the whole thing again. The main things to note for this are, you use the light reactions in your thylakoid membrane. You're going to break water to get that electron to go through the electron transport chain. When you break water, you're going to release oxygen. That's a waste product. The energy molecules you make in the light reactions power the Calvin cycle. The Calvin cycle fixes carbon dioxide from the atmosphere into an organic molecule that you can turn into sugar. So if glucose is a 6-carbon sugar, that carbon in the glucose comes from the air. We're literally inhaling it. If you were a plant, we're exhaling it because for us it's a waste product. Cool. So here's a practice multiple choice question for you. The process of carbon fixation is important because it converts and if you said E, carbon dioxide into sugar, you are correct. Fixation means to take something that's inorganic and make it organic. The carbon dioxide in the air is inorganic, and once it's in the form of sugar, it's organic and we can use it. What color light is used for photosynthesis? If you said all colors except green, you're correct. The photosynthetic pigments absorb most colors, but don't really absorb green, and so green is reflected and refracted off of our leaf, making the leaf appear green to our eyes. So I asked in person, what's a plant's favorite color? And you want to say green because they look green to us, but really they use all the other colors to make food, and so that would be their favorite colors, everything except green. And then the last thing I have for you for this lecture is a study suggestion. So I recommend doing a concept map for this. The end of the term, we're kind of throwing bigger vocab words at you and more complex processes. And a good way to organize your thoughts and figure out what you need to study more is to do a concept map. So pause the video here, make a concept map. Remember that you need to connect your concept words, your vocabulary, with some kind of description or phrase so that you understand how two things are related. So like I might do the electron transport chain and the light reactions, which are not on here, but that's okay. I can add things. I could do the electron transport chain and oxidative phosphorylation. So whatever you need to do to connect these words appropriately, go for it. And definitely add more in. This is just a bare bones one to get started. And don't forget about our course study guides. So those are up on Blackboard. Fabulous. Thank you so much for this lecture, and I hope you have a great day. And as always, email me, please.